We'd like to continue now with the presentation of what would be what we call the regulatory perspective on the problem of SMA1. I'd like to introduce Dr. Haberkamp, who is a clinical assessor at the BfArM. She's also a member of the CNS Working Party and the Scientific Advice Working Party, and she'll be delivering a presentation on the regulatory perspective in SMA1.
Thank you very much, Pavel, for this nice introduction. And hello, everybody. Well, the scene is already well set. From the lectures this morning, we heard a lot about the clinical picture of the spinal muscular atrophy's disease spectrum, about the genetic background, about therapies who are being developed, about endpoints, biomarkers, and natural history courses. And it is my aim now in the talk to give you a regulatory perspective on strategies for drug development in spinal muscular atrophy. And I will concentrate on SMI type 1, since SMI type 2 and 3 will be dealt with this afternoon a little bit later. This is my disclaimer, and this is an overview of the contents of my talk. I would first like to start with the key characteristics of the type 1 population, about the appropriate outcome measures. Then I would like to elaborate a little bit about trial design features, and I would like to close with an outlook, what we can offer as regulators, and critical issues for discussion. Well, this is already a recapitulation of what we heard, a summary of the most typical clinical characteristics of SMA type 1. And I would like to highlight what is important to regulators. Type 1 is the most severe and common type, which accounts for about 50% of patient cases, as we already heard. The onset is typically before 6 months of age, and we have a high mortality within the first 2 years. However, survival has improved in the last few decades, and this is due to improved standard of care, including respiratory techniques, nutritional techniques, but also cough augmentation techniques, and this has to be carefully taken into account when we plan clinical trials. The selective degeneration of alpha motor neurons and the ventral horn of the spinal cord and the brainstem leads to the typical clinical picture of the floppy babies. And we heard that sometimes the disease is further subdivided in severity stages. However, all stages have in common that patients are never able to sit, and this is the motor milestone they never reach, and the weakness they have and the other complications lead to lots of complications, the risk of aspiration pneumonia and respiratory insufficiency and fatal weakness. We saw already this slide, but I've also put it in from the publication of Kalb and Kissel because it nicely depicts the timeline where we are right now and where we started. The disease was first described at the end of the 19th century by two neurologists, the Austrian Guido Wernicke and the German Johann Hoffmann, and it took 100 years to detect the SMN gene and 20 years now to develop the animal models to test potential therapies. And we are right now in a situation where we have a silver lining on the horizon. Several drugs are tested in several stages of clinical development, and in this situation that we have on the one side the high unmet medical need to reverse, delay, or halt the progressive decline in spinal muscular atrophy, and that we have emergent treatment options, we need regulatory standards to evaluate efficacy and safety of a prospective treatment and to establish benefit risk in a very vulnerable patient group. And we have really to show that the efficacy is not due to external factors, that is really efficacious, and that the treatment is safe. And as regulator, we are well aware of the challenges and of the ethical considerations with respect to prolongation of suffering when treatment is started too late in SMA type 1. And we are also very aware that parents and carers always have the choice between palliative care and intervention, and that should be carefully considered when planning to do trials with those patients and during the trials as well, of course. Well, this is the clinical disease classification, which we already heard about, classically type 1 to type 4, and there's a further type 0 in the most extreme margin. It is based on the age of onset and the highest function achieved. This classification is certainly very helpful for clinical reasons, but it might not be that adequate when it comes to prognostic information and for the stratification of patients into clinical trials. And we are aware of the fact that the disease exists as a spectrum with a continuous range of severity, and then therefore for the purpose of clinical trial planning, it might be more appropriate to alternatively classify between early and late onset SMA. The cutoff point is usually set at 6 months. This might be debatable, but however, it nicely fits to the SMA type 1 spectrum onset of efficacy. And it is always easier to show an effect in a homogeneous study population, and that accounts for the patients who are younger than 6 months of age. However, patients older than 6 months of age in this case will be very heterogeneous and need to be characterized otherwise, and we will hear about this afternoon. And that immediately raises the question about extrapolation. How far can we data in SMA type 1 patients extrapolate to other stages? And I will come back to this a little bit later. Well, what is the typical type 1 SMA patient? How do we define the inclusion criteria? We already heard about that. It should be a genetic confirmation of a homozygous loss of the SMN1 gene. It accounts for 95% of the cases. The other 5% are rarer mutations and compound heterozygosity. They should not be excluded from the trials. So we encourage to include all those patients in the trials. And patients should have the clinical typical history with an onset of symptoms prior to the age of 3, and the inability to sit and independently. They should not have reached the motor milestone sitting. And the SMN2 copy number is important. That should be established, and usually 2 copies are required, and this is nicely supported by a recent publication from the Neurodex data group in 2016, which had a retrospective look at 26 SMA patients, and all started before the age of 3 months, and all had 2 SMN2 copy numbers. So we can be confident that we have a homogeneous population there. We are aware that we have a narrow therapeutic time window. Efforts should be undertaken to enroll patients as soon as possible after diagnosis, and ideally prior to onset of significant degeneration, so that the therapy can exert its effect. But how early is early? Does it mean identification of early symptomatic or even pre-symptomatic children? And this is also a challenge. It might be easier in families where siblings are already affected to do genetic testing, but then when it comes to newborn screening, this is seen more controversial still, and it might be discussed otherwise after the approval of first effective treatments. Until now, we don't have any cure for SMA. However, we have standards of care, a consensus statement published in 2007 by the International Consensus Group on Standard of Care in Spinal Muscular Atrophy, and this is widely implemented, as shown by a UK group in 2011. But on the other hand, as published by the MDA study group, which mapped the differences and had a look at 24 national registries across the world, there are quite a lot of differences in standard of care. And when it comes to invasive ventilation, for example, it is more aggressively handled in the US than in Europe. Nutritional support is more aggressively handled in Europe than in Western Europe in comparison to Eastern Europe, and other aspects are also different. So this has to be carefully taken into account when it comes to trial planning and when we look at the primary endpoints. Time to event endpoints, of course, survival and other end-of-life measures, such as time to full-time ventilation, however it is defined, are important outcome measures. This is clear. However, they are highly variable due to variable standard of care, and either we have to homogenize that for the study inclusion criteria, which might be ethically difficult, or stratify for this, which might also be difficult because we have not so many patients to stratify. Not all SMA type 1 patients are available for clinical trials, and companies are also competing for the development programs. So that is difficult. So that's why we think that gross motor function milestones, such as the proportion of infants sitting after 12 months as primary endpoint, might be preferred. And that is supported by a recent publication by DeSantis in this year, which showed that despite standard of care, improved standard of care, all of the patients that they had a look at as spinal muscular type 1, they never achieved the motor milestone sitting. So this is really a valuable endpoint, and there are scales that can be used, like the Heine, as we already heard in a recent talk, and the Bailey infant motor scale to establish those motor milestones. However, it should be standardized. Sitting without support for how long? 5 seconds has been suggested. That could also be debatable. However, and it should be video recorded. We recommend that in a standardized manner in clinical trials and centrally reviewed, and ideally by independent raters. And the effect should be supported by a consistent effect in secondary and further endpoints. Survival and end-of-life measures could be a key secondary important endpoint, but also the more continuous motor performance scales, respiratory function, which is a challenge in very young children. We can't take the usual parameters, but there are alternatives like the inductive plethysmography or the improvement in asynchronity between diaphragmatic breathing and thoracic cage-driven breathing. Quality of life measures are an important outcome in clinical trials and should be measured. I'm not aware right now if we have specific scales in spinal muscular atrophy. I think they are in development, but we have generic scales that can be taken. Caregiver burden should be established, growth parameters, weight gain, and of course the electrophysiological measures the biomarkers we heard about. We have a wide range of motor performance scales that can be used and had been used in SMA type 1, and we had a nice overview on that. They all have strengths and weaknesses, and as regulators, we are not prescriptive in what scale should be taken. The CHOP intent has good intra and inter-rater reliability and has been widely used in clinical trials, but other scales could also be used. They need to be justified, and the clinically important difference need to be established and mentioned before for the trial planning. That's important, and we will always have a look at the whole picture as regulators in case the primary endpoint not necessarily gives an improvement, but a slowing of the decline. Those secondary endpoints are really very important. Now we come to trial design. What is the ideal trial? Of course, a placebo-controlled study would be the optimal design to control for all kinds of confounding factors. That is always the case. However, in the specific case of type 1 SMA patients, regulators are also open to open label designs versus historical controls. And this is not only because of the rapid decline and the low life expectancy, but above all because those patients never achieve the motor milestone sitting, as already mentioned and shown by DeSantis in 2016. So when we rely on historical controls, we need an endpoint which is not subject to bias, and to sit and not to sit is not really subject to bias. So we can have a great confidence in that. And we now not necessarily need an internal control arm. But this approach should be carefully planned and discussed with regulators, and usually patients should be carefully matched if we do this comparison on a patient-by-patient basis. This might be exactly the difficulty, especially for the very young patients, which are unfortunately underrepresented in these national registries. So we have a little bit caveat here for the very young patients. How long should be the study duration? It depends on the mechanism of action and the endpoint chosen. And in case modification or slowing of disease progression or even improvement is shown, we require usually 12 months data at least. And this nicely fits also to the motor milestone sitting, because all patients should have achieved normally sitting by the age of 12 months. The maintenance of effect needs also to be assessed. How is this ability to sit acquired once for all, or is it only futile? And so we need long-term data to establish the maintenance of effect with an additional observation period, and after marketing authorization, also the observation of those patients in registries. How many trials are needed? Well, this is an orphan disease scenario, and we are open to the fact that one well-conducted study could be sufficient for type 1 SMA. However, the data should be sufficient to allow benefit-risk assessment. And we think that a second study across the disease spectrum will be needed in case it should be given to also to other patients. And this is because of older patients and younger patients have differences in physiology, in metabolic pathways, and in different profile of comorbidities, which might influence the effects of drugs used. And this should be carefully evaluated. And therefore, we need also distinct developments and studies in older patient groups. This is about the role of biomarkers. We already heard a whole talk about, and I don't want to insist to recapitulate the role of biomarkers, but we have, of course, the electrophysiological biomarkers and the muscle mass quantification biomarkers, which are also coming into place, and the molecular biomarkers and the protein levels. The electrophysiological markers could be used as a biomarker of change in neuromuscular function, and the SMA transcripts and protein levels can be used to indicate pharmacological activity of a drug. But however, as we already heard, the surrogacy is not established yet, so we can't rely on those those biomarkers as primary endpoints right now, but they should be included in the trials as supportive endpoints to complete the whole picture, and we need to identify and validate additional biomarkers and validate the existing biomarkers we have. And what can we offer here as regulators? Well, we can always offer scientific advice and protocol assistance, and when it comes to the biomarkers and certain endpoints, we can even give qualification, opinion, or advice based on the acceptability of specific use of a proposed method in a research and development context, and we will look and assess the data or the preliminary data provided to us. And EMA is also dedicated to expedite marketing authorization when it comes to approval of drugs. And we have several tools here. We have the conditional marketing authorization in case we have an unmet medical need and seriously debilitating or life-threatening disease, as we have in SMA. And this should be carefully planned. And early approval on the basis of less complete data does not mean that the benefit risk does not need to be established on these data. We have the tool of accelerated assessment with a shorter assessment time, 210 to 150 days. And we have newly established in this year the BfArM scheme with an early dialogue at the time already of proof of principle between scientific advice working party and CHMP regulators and rapporteurs, and a new scheme for unmet medical need and major public health interest is really a nice opportunity to accompany the development program and give advice throughout the development. And I would like to close now already with my main issues for discussion, which might be helpful to discuss later. What is the ideal definition of populations? How early should we include patients into the trial? Early diagnosis versus pre-symptomatic or newborn screening. The importance of registries and the improvement of registries in the very young has to be stressed. We need validation and identification of biomarkers. The question of extrapolation needs to be more elaborated, and how long should the maintenance of effect be established, and what will be the scenario, lastly, in case of approval of one drug. Do we need then a comparative superiority trial, or is it also appropriate to have then a placebo-controlled trial, or still a control versus historical controls? And with that, I would like to thank you for your attention and close my talk.
Thank you, Dr. Haberkamp. I'm sure that this will raise a lot of questions in the discussion session that comes later on, and I also have to remind the participants that we have a large participation of regulators, except from the ones that you see at this side. On the other side, we have assessors from national agencies. We have the chair of the Scientific Advice Working Party and colleagues that are dealing with the topic, which I believe would also comment when the time comes. But for now, in order to present the industry perspective on the same or similar topics and difficulties in SMA1, I'd like to introduce Kathie Bishop, who, despite not being part of SMA development right now, from what I understand, had a very leading role in previous life and has a great experience in SMA1. So Kathie,
Thank you.
yes.
Yes, so my disclosures are that I currently work for a company called Tioga Pharmaceuticals, but I'm not involved in any way with companies, and I don't have any financial relationships with companies developing drugs in SMA. From 2009 to 2015, though, I was a full-time employee of Ionis Pharmaceuticals, where I led the development of the SMA program from pre-IND through design and initial enrollment of the phase 3 studies, both in type 1 SMA and later onset SMA. And actually also prefer the terms earlier onset and later onset SMA in terms of classification. So I really want to thank the EMA and SMA Europe and Treat MND for organizing this meeting. Drug development in SMA is very difficult, mostly due to the heterogeneity of the disease. I think in type 1 SMA, it's even more difficult because of the very severe and rapidly progressing nature of the disease. And you heard from Elliot, I think this morning, a little bit about what that's like. I think it's made possible, though, because there is such great collaboration within the SMA field between physicians, industry, physical therapists, and regulators. And that really, this meeting helps that along as a really important forum for sharing experiences and viewpoints on this task. So thank you. So, and I will say also the views I'm going to express are my own. I've tried to think about this, though, from an overall industry perspective. As was presented this morning, there are 6 drugs now in clinical development in SMA. There are more drugs also in preclinical development, which I think is very exciting that there are so many approaches being taken. Some of these are direct SMA replacement or SMA production therapies, but then also others of these are different mechanisms involving the muscle and different mechanisms. And I would like to see a future where there are many drugs for SMA and combinations of therapies, because I don't think one drug alone will solve all the problems in SMA. So I also, as I went through, tried to think about the overall drug development process. And so these are the topics I'm going to touch on briefly. I will say, too, being in industry, I consider it industry's responsibility to provide clear evidence of efficacy and safety that then the drug regulators can evaluate. But it is our responsibility to provide that data, to design trials that will produce a clear result, to conduct the trials that will produce a clear result, and then to provide clear data to the agencies. Sort of balanced against that and some of the challenges in type 1 SMA is that it is a very heterogeneous disease. It's a very small patient population. It's very severe, very rapidly progressing disease, which makes it very challenging conducting trials. There's variability in standard of care, and then there's also potential ethical issues, and I'll touch on some of these points as I go through. So as I mentioned, the first topic is patient population. The reality is that type 1 SMA is a very severe, rapidly progressing disease with permanent ventilation or death eventual outcome. And that makes conducting clinical trials challenging. I also think one of the things Elliot mentioned this morning is really important to keep in mind is that parents and patients with type 1 SMA already have a substantial burden. And for them to enroll in a clinical trial just produces more burden, especially when you're thinking about the types of outcome measures, the visit schedule, how many outcome measures that really needs to be carefully considered. I think, as Elliot mentioned, just conducting the CHOP INTEND test alone is a burden on these children. So if you're thinking about multiple outcome measures, that really needs to be thought through carefully. It's also a rare disease. If we're thinking about enrolling patients early in disease, then I've given some numbers here on the estimated births per year. This might not be accurate. It might be a little bit more than this, but just to make the point that there's very few patients born with SMA each year number in the hundreds. And so for enrolling trials, we have to think about the size of the trials and that carefully. And then, as I mentioned, it's a heterogeneous disease. As other people presented, the copy number is a really good way of producing a more homogeneous population, limiting trials in type 1 to copy number 2. Not to say that patients with copy number 3 in type 1 SMA wouldn't also benefit, but just to produce a more homogeneous population in which to prove a drug effect. However, even within copy number 2, there is unexplained variability both in disease onset and disease progression. Quite a bit of variability, actually, that's not yet explained with other factors and doesn't correlate with other factors. So you're still left with some heterogeneity. And then, as was mentioned, I think standard of care and variations in standard of care are very important in type 1 SMA. If you're doing global trials or trials within multiple countries within Europe or other regions, there's variations country to country, also investigator to investigator, site to site. But then there's also, for some issues in standard of care, especially ventilation, BiPAP, tracheostomy, there is parental choice that is just part of the disease. And so this also compounds the variability and has to be carefully considered. I think it's fairly well accepted now that treatment as early as possible is likely to provide the best potential for benefit. And so when thinking about a clinical trial in type 1 SMA, enrolling patients as early as possible gives you the greatest chance for success. It's been established even at symptom onset, when these babies first present with symptoms, there is already pretty significant neuronal loss and degeneration. And then progressing from there, it's very rapid progression. Depending on the mechanism of the drug itself may require some time for an effect, which also makes it enrolling early important. However, limiting criteria to early enrollment also then limits the ability to enroll the study, so it has to be balanced. Oops, okay. So then I turn to the topic of control group. And the control group chosen may depend on the size of the drug effect expected, whether it's a disease-targeted SMN replacement strategy versus maybe in the future symptomatic treatments to be added on top of that, and the choice of endpoints used. Placebo or sham-controlled trials can be conducted in type 1 SMA, and one has been done, phase 3 trial, so it is possible. To help with the ethical issues with that, we recommend an imbalance in randomization, more patients on drug than placebo. However, historical control trials could also be conducted. It's important, though, to think about that the historical control matches the treated trial in terms of standard of care. There's a contemporaneous study. There's pre-specified. And I will be a little bit controversial in asking whether in Europe there are robust multi-center studies with current standard of care that would meet these definitions. And I think that's an item for discussion. As Richard presented, in the US, there's been 2 recent multi-center studies, the PNCR study and the NeuroNext study. But as he presented, the European studies, I think, are a little bit older than that, limited to 1 country in most cases. And the standard of care might be more variable. So in some cases, there may be ethical issues with conducting a placebo or sham control randomized trial, but that needs to be balanced with the need to provide conclusive evidence of efficacy. And thinking about other potential trial designs, because of the severe rapid progression, it's not conducive to crossover or delayed entry studies because the patients do progress so rapidly. And an option where you might consider providing a rescue treatment when patients reach an endpoint potentially may create bias. So that's something also to think about. Open label extension studies are, of course, optimal. And then I think one thing that's been mentioned here and there this morning is really making the studies as short as possible. And using the creative use of interim analyses to limit the duration of the trial or the number of patients that may need to be studied to get to an answer as quick as possible because of this severe, rapidly progressing nature of the disease. So my outcomes study looks very much like Marion's, which I think is reassuring. We didn't work together on this. And I will start with the outcome measure of time to death or a surrogate of permanent ventilation, which has generally been defined as greater than or equal to 16 hours a day of ventilation for at least 2 weeks, or I think in the Ionis trial we used 3 weeks, in the absence of an acute reversible illness or tracheostomy. And I will say, in practicality in implementing this endpoint, it's not an easy endpoint to implement. When you start to think about all the components of this endpoint, it becomes complex and difficult. It needs daily diary, and maybe even adjudication when the endpoint has been met. If you read the definition, it's very complicated, and it's not easy to see when they're past 2 or 3 weeks and whether they actually have an acute reversible illness or not. And it's something, when we're providing data to regulators, we need a clear answer on that for them to evaluate. It also is affected by standard of care differences in respiratory use, particularly BiPAP, and whether parents would choose tracheostomy or not. And I think also in SMA, the respiratory progression is not a nice, smooth slope rate of decline. It's a little bit of a random event. SMA infants can acquire a respiratory infection at any time, and that really produces a respiratory crisis, which is when they would require sort of run into where they're going to meet this permanent ventilation endpoint. So there is a randomness factor to this endpoint. And this endpoint may also require larger sample sizes and longer trials. So I also prefer the use of motor milestones. Sitting by definition is not achieved in type 1 SMA, so that's, I think, a clear outcome measure. There's other motor milestones, though, such as rolling, standing, other things that should be looked at as well. The Heine-2, as Basil presented this morning, which is the Hammersmith Infant Neurological Exam Part 2, I think is very nice in that it looks at these motor milestones, but it does so and allows incremental improvements to be scored. So you can look across a range of motor milestones. You can look for smaller improvements, not just have they achieved the ultimate milestone or not, and you can generate a score. And I think that's a useful thing to do. Motor milestones also, I think, offer the advantage that they may require shorter duration trials and fewer subjects. There has been maybe some movement to thinking about a co-primary, having both outcomes, which I think is difficult because it does require more power and a greater number of subjects to have 2 primary outcome measures. And in the small patient population is something that I don't think is helpful. So in addition, there are other motor function scales that can be thought about. The CHOP-INTEND was presented. It was developed specifically for SMA infants, and I think is a really good motor function scale in SMA. And then there's more general infant scales that Basil also nicely presented this morning, such as the Ames and the Bailey-3, that can be considered. And then other measures of impact on disease, such as disease-related adverse events and serious adverse events, hospitalizations, G-tube use in feeding, which is very important, and time to requiring BiPAP. Although some of these, such as G-tube use and time to requiring BiPAP, again, may be biased by care differences and differences in standard of care. So moving to biomarkers and patient-reported outcomes, Arthur did a very nice job this morning of summarizing the work on the biomarkers. I would say that CMAP, I think, is emerging as a nice biomarker. It correlates nicely with motor function. Traditionally, CMAP of the ulnar nerve has been studied in SMA type 1, but we've also collected data on the perineal nerve, which I think offers the advantage that at least at baseline, it is less deinnervated. And so maybe there's a greater chance to show a response using that nerve. So I think both can be considered. Muni is not generally feasible in SMA infants, and I think adds a real burden, so would not be recommended. CSF and plasma biomarkers can be considered, which I think Arthur very nicely summarized. For some mechanisms of drugs, direct pharmacodynamic evidence and PK/PD analysis can be done with these. But it should be remembered that peripheral biomarkers do not reflect target CNS motor neuron changes. And then Arthur nicely presented some exploratory biomarkers that could be considered, but there aren't any established right now for clinical trials. Also should be considered from a practical perspective that in type 1 SMA infants, due to their small body size, you're very limited when you conduct clinical trials in the amount of blood that you can collect, especially at baseline when they're small. And so there's not a lot of room for exploratory analysis, and I think that's important to realize as a fact as well. PROs, in this case, they're not patient-reported outcomes, but they would be parent-reported and or physician-reported. And I think as someone mentioned this morning, there are none developed specifically for SMA, but more general scales could be adopted. I also want to bring up, which I think is could be an uncomfortable topic, but I think is very important, that inclusion of autopsy analysis should be considered in clinical trials of therapeutics. They can provide invaluable data, especially on the mechanism of action of the drug and PK/PD analysis, and that's a way to collect data directly on the CNS if the opportunity provides, and it should be something to be considered. So as I mentioned, there are the challenges of conducting these trials. I want to touch on one briefly, which is standard of care. So in conducting trials in type 1 SMA, there's advantages to standardizing standard of care. It reduces the variability, and it also makes sure that the patients are receiving the minimum care standards, especially with regards to nutrition and hydration. Drugs may take some time to have an effect, so it's important to make sure that the patients meet a minimum standard of care. It also could be possible that drugs may have a greater effect when patients are receiving better standard of care. And so I think it's important to make sure that the standard of care is there. Richard and myself and Skip Nelson recently published a paper on the ethics of standardizing care in type 1 SMA, because it is a difficult issue to think about. As I mentioned, because these patients are so very, very fragile, it's important to think about the visit schedule and the assessment burden and minimize that as much as possible. Also, it's important to realize that most of these babies will travel to a study center, so that needs to be taken into account in minimizing, I think, the visit schedule and spreading visits, as Elliot mentioned, out over 2 days rather than 1 day to make it more comfortable for the patients. And then, as Marion mentioned, I think one question is extrapolation of results to broader patient populations. With these challenges, what we end up doing is restricting our patient population in our clinical trials. But hopefully, people realize that hopefully these results should extrapolate more broadly, not just those meeting the inclusion-exclusion criteria. And I think that's a good point for discussion, not only in type 1 SMA, but also in type 2 and 3 SMA. So also, as we think about the future, industry, I think, also has other things on their mind as well, especially some of the companies coming along. And what will the impact be of potentially changing phenotypes of type 1 patients who may be treated with therapies that may be approved in the future? Will we then, for follow-on treatments, have to conduct comparative studies or combination studies? What will happen with changing standard of care? And I think even increasing the awareness of SMA and with drug trials being conducted, there is hope on the part of parents. And I think that hope also impacts the standard of care. And then, as Marion mentioned as well, with an approved therapy, will newborn screening eventually be implemented, especially here in Europe, where I think it might be a little more challenging with the country differences. So thank you.
Thank you very much for the overview and flagging out these important questions. And I'd like to actually give the floor now to Drs. Finkel and Tiziano to lead the discussion, and maybe we can use some slides to visualize it better.
Hello, good morning, everybody. I'm very happy to be here. I'm Eduardo Tiziano. I'm director of the area of clinical genetics and rare disease unit in the hospital BioFArM in Barcelona, Spain. I've been working in SMA during several years, and this is a great moment and just an historical moment to have here just all these people devoted to the disease in different ways. Just to facilitate the discussion and questions with Richard Finkel, we are going to facilitate starting with some questions that could be probably open to the audience. That's ethical issues of placebo-controlled studies in type 1 SMA. I have here from Elliot this morning that says we have been taking placebo for many years in such a way, in the ways as to decide what could be the parents' decision about the questions to be included in a study with placebos or not. And the second one is the use of palliative care in studies in type 1 SMA. So we have very wonderful discussion this morning on this one. And so it's really the third alternative is just to have open questions from the audience during all the sessions that we have had before. So please, we want just to start with the ethical issue. If somebody wants to make an opinion, questions, or discussion on that. And after we reopen just to try to make a discussion on all the questions that appears from the audience. I have one question there, please. Can you present yourself and please be
Joseph Irwin, I'm from SMA Support UK, but I'm also on Treat NMD ethics panel. And this is a major issue for our families because everyone, and it came out in a number of the histories today, it's really the problem of joining the studies, the fear that you're going to go onto the placebo arm. And as done in the regulatory presentation, we now have quite a lot of natural history data. It's fairly recent natural history data, where before it was older information, and we also have comparative studies. So the question is it really fair in a type 1 group where the outcome is death to actually use placebo studies?
Somebody want to answer or to help in the answer? Just let me go to the next slides. So just starting to try what could be the utility of placebo, because provides natural history in the context of a trial. That this is very important for clinicians. So we probably, in a disease like type 1 SMA, we have to rely on natural history studies that have been done before. So we know that a patient under a clinical trial have a different type of care and a different from the family and from the medical staff. And of course, to define the limitations that there are in natural history studies, not in the context of the comparison with a drug that is under study. And the other question is, when is a single arm study appropriate? So when we follow this, we have a drug under investigation, the drug under investigation with convergence of multiple studies. So we are probably have the possibility now in this scenario right now. So we will have probably more than one patients to be decided to enroll in one clinical trial or in another clinical trial. And of this will make recruitment challenge. And of course, if we have the effective drug, we will have the impact on the field. Because if we know there is something is effective in the disease, we should have to take care with it. We change probably, and we will influence the standard of care. And of course, future clinical trial design. As Kathie just mentioned, the possibility to make comparative studies instead of doing really placebo studies at the end, or just to have combinational studies in the future. I don't have a specific answer from you, from our side. This is probably one of the answers why these placebo studies are very important in the definition of, or in the design of a clinical trial. But Eugenio probably can have.
Well, I wish I had. No, I was very impressed and very happy to hear from the MA, from Marion today about the possibility that the placebo may not be considered if there is the achievement of major milestones like sitting, which is something that I'm sure will be appreciated. However, achieving sitting is a major achievement, is raising the bar very high, because this means that these children are not type 1 any longer, they become type 2. And we may have some drugs which may be effective, but especially if they work on children who are more advanced in the disease, who have less motor neurons, we may still see an improvement in other milestones like holding the head, having some trunk posture, reaching with hands and so on, that they may not achieve sitting, but they may still achieve many other important milestones which are in between from being completely flat and not having any movement and sitting, which are in between. So I was wondering whether in this case placebo should be needed or not, because this is an intermediate situation which is probably more likely to occur in the older children.
So I can try to reiterate what was presented by Dr. Haberkamp, and also I'd like to invite our assessors on the other side of this room to take their position as well. But I think this was used as an example and nothing more. The position as presented was that milestones are better for the reasons that we saw. And of course, if you make the case that a certain milestone is achieved and is important for the patient, it doesn't have to be a major, as you call it, milestone, transferring it or converting into a curative effect. And I think this became also clear, but just to reiterate, we understand that maintenance of function is sufficient. And because it was mentioned sometimes as an improvement, we don't necessarily need to see an improvement. Maintenance of effect is also achievable, is also an endpoint which is sufficient and enough for us. But I'd like to hear also the points from our side. Andre?
Yeah, Andre Alfred speaking from the Dutch Medical Evaluation Board. This is an orphan disease, and for the planning of your studies you need a primary endpoint, and the primary endpoint of milestone is usually clinical relevant. But we are not only looking at primary endpoints, we also look at the secondary endpoints and the consistency with the primary endpoints. In that sense, if you see a large effect on one endpoint but not on the others, even not in direction or whatever, there is something there's something questionable. So we can focus on a primary endpoint in the sense of, yes, we need it for planning the study and preferable it's a clinical relevant endpoint, that sort of things. But we will look at the whole picture, especially in the context of an orphan disease. If it's not a well-established disease like rheumatoid arthritis, where you know that a primary endpoint represents the whole clinical picture, that's the ACR20. These are all snapshot pictures. And of course, the primary endpoint is important for your planning, and the primary endpoint can be relevant, but we will look also at other functions. And you should keep that in mind. It's not only, well, if we meet the primary endpoint, then we have a success. No, you should be consistent.
I'm really totally agree with that. The thing is, why we could change the answers of the families in the context, because sometimes we see just in the following that is the drug is really the outcome is really good. So probably we are knowing that this patient has something different from the natural history, and that would be what could be really the decision on continue the treatment under the placebo or not, just to make a decision like an interim analysis that will make really things going faster and decisions to have all kids on open label. You want to try?
Well, doesn't this just depend on the degree of effect? I mean, we're back to the same thing. If it has a huge effect, then you don't need a placebo arm. If it has a minor effect, I'm not saying it's not significant, then you'll probably need a placebo. I mean, I would go back to your comment that you were making about, do you need to do another natural history study in Europe, given that there's been 2 done in the US, and there's no statistical difference, like for instance on death, between the older European studies if you include the time to ventilation. To me, that means that you don't need to do a natural history study in Europe or have a placebo control if you have a large effect.
For me, it seems extremely important to discuss the issue really whether the patient you include is a kind of naive patient, so it didn't get any drug before, or it had another drug before, and then you have to consider the milestones this patient has by the time you include that patient. And I think for type 1, indeed, you can consider only a kind of naive patient really to have a type 1 SMA patient, because if that patient has been already on another drug, then it's getting very difficult to still keep considering that patient a type 1 SMA patient. I think that is something which I believe has to be taken into account also when we consider whether we should go for a placebo or not.
Yes.
So it's more to reiterate, our problem is we see drug A, placebo control or treatment, drug B, placebo control or treatment, drug C. But what we have as a community is a database of children where they have either received no treatment or treatment A, B, or C. And what our weakness is, we are using a very old model in a small cohort of patients, and we are necessitating a control again and again. And I think that's the ethical issue, because we should have access, not maybe it has to be something like in the agency, but there should be a way that you can use the data that's known to compare any other new treatment that's being considered. And that's the issue, we think, from an ethical point of view.
If I could just elaborate on what Eugenio brought up before. So if we have a drug that shows what we feel is some response, but you're not achieving a new milestone, would a motor scale, such as CHOP INTEND, be sufficient to provide convincing evidence to the regulatory authorities, particularly if there's divergence? If are we convinced that there's enough natural history data that in type 1 infants there's a decline, and if you have a drug that shows an improvement on a scale, albeit without an improvement in a milestone, you're not gaining a new milestone, would that divergence in a scale be sufficient to warrant drug approval?
Yeah, yes, I mean, I would share the view of the fact that finally the need for a placebo relies on the effect size. A placebo, why is needed a placebo? It's needed to ascertain that the clinical effect that you see, they are actually due to the treatment. And when the effect size is striking, where they never sit and now they sit, you don't need a placebo, you even don't need a control, you know the natural history. But it's in the case you have a halting of the disease progression due to the treatment. And we cannot take that for granted. And in a number of cases, all the treatments, they only are going to only slow disease progression. And there you will need some sort of a comparison. You will need controls, because how assessing disease progression, halting on disease progression. But in SMA, we also have to take into account that if we have a very rapid disease progression, if the halting is sufficiently important, you could. But I mean, really, it's it's an issue of effect size.
Yes, and to come back to your question, if the top intent would be acceptable as primary endpoint, well, it depends if a clinical relevance can be shown. What does the effect size you show mean for the patient? Is it only statistically significant, or is it also clinically relevant? And then you need to make a case when you plan the trial to define that a priori and to back it up by ideally secondary endpoints and the mechanism of action. And so we don't say no for that type of endpoints, but still it has to be carefully planned, and the effect size it has to be shown that the effect size is really meaningful for the patient and not only a statistical value which might be very slow, but very low and does not mean anything.
So to try to probably summarize what the regulatory point of view here is, we are not requiring as such a placebo-controlled trial, and this is to explain to the patients we need that the data are good, and there are ways to make data good even with a natural history study, and they were mentioned in Dr. Haberkamp's talk. You have to make sure that the patients are properly matched, that you have identified them properly, that you're measuring the same thing, that the standard of care is such that you can rely that there's going to be no effect of the standard of care that was applied in the natural history study and the one that you're applying in your study on the potential outcome of the endpoint. Provided all of these are matched and true, we are not, as regulators, requiring that a placebo-controlled study is done, but that this has another side of it. And I would like to hear the industry position, because in the planning, as mentioned by Dr. Elfering, when you're planning for your studies, you may believe very much in the effect of your drug, but you don't know it. So are you going to take the risk of having a natural history control, and potentially miss out on a good drug that is showing a less than expected efficacy? Or are you going to take the risk of having a placebo-controlled trial and miss out on the fact that patients may not be recruited and all the other potential issues that come with that? So I would like to say it's not us driving the decision only. There is another side, and Kathie, maybe you can have a position, but I would also like to hear the other representatives of industry on that.
Yeah, I'll respond, but I would like to hear from other members of industry as well. So I agree absolutely with what you said, Pavel. And I think that was just to share a personal experience, when we designed the phase 3 trial for Ionis, of course, we discussed at length whether it was ethical to do, in this case, a sham control trial, but it's equivalent to a placebo control trial, or whether we would do open label compared to national history. And it's exactly the point that you make, is our obligation is to provide clear evidence. And because I think, especially because this was the first phase 3 trial in type 1 SMA, we did not know in the end what the size of the drug effect would be. So if we would have gone ahead and done it against natural history, we may be risking that we would do the trial, not have a clear answer, and it would all be for nothing, including the sacrifices of the parents and the patients who were in the trial. So we did choose to do a sham-controlled trial, even though I think it's ethically more difficult, but because we wanted at the end to have a clear answer. And I think that's from the data so far, that's what happened. And so that was the thinking behind that choice, and it's exactly what you're talking about, Pavel. I think more generally, the choice of what your control group depends on what your drug is, what the size of the effect that you would hope or expect to have, what endpoints you're using. You can do placebo-controlled trials, you can do natural history controlled trials, but you have to make sure that whatever the control is meets the standards for having a clear answer at the end of the trial. And things like Pavel mentioned, if you're going to do a natural history control, making sure it's contemporaneous, meets the same standards of care, patients are matched, and things like that also needs to be considered. So, and I invite, yeah.
Yeah, I was thinking that when considering the trade-off between the need for a placebo and the recruitment difficulties, I was thinking, for example, a drug that would delay the time to full ventilation, or that would decrease the need for the number of hours on ventilation during a time frame, and you would really need a comparison. perhaps to fill this gap and to find a trade-off, we should include in our reflection consideration on alternative designs, taking into account the very restricted sample size, trying to find an optimal minimal duration on placebo, alternative designs, introducing some randomization, some comparison, but perhaps with less reduced time on placebo when compared to the classical parallel design.
Yeah, and that's exactly the discussions that we had with the design of the trial, is having an interim analysis as soon as was statistically possible in order to reduce the time on the trial, randomized 2-to-1. So agree completely, thinking about novel trial designs to address these ethical issues.
Before, I want to ask you, if we have a good natural history in the placebo arm, in a clinical trial, can we define this for the future, and we can use that? Because this, if we are going to follow in the same standard of care in new designs of the trials, perhaps we can use that one, and we can modify the design taking into account, or how is this one?
Julie.
I just wanted to say that I'm not sure that an interim analysis will provide the requested level of proof. I was more thinking of alternative designs such as crossover design, delayed start design, randomized withdrawal and re-challenge design, all those sort of things.
Yes, Sylvie Benstreet from the PDCO. The alternative designs like adaptative or N-1 could be an intermediate solution to have some standardization and to prove something, but also to decrease the timelines. And what is important also in addition is to consider interim analysis regarding the dose issue, because more than one time it could be indeed challenging to assess the good efficacy safety ratio, also to assess after the benefit risk of the drug. And interim analysis could then solve in a small population before to engage more patients and children, in particular on longer clinical trials.
Sorry, we're monopolizing the discussion from this end. My name is Angelica Siapkar. I'm the UK PDCO delegate. I think first of all, this is a great meeting, and I think it will be helpful for everybody, and it's very good to hear patient views on this very important subject. I think the landscape is changing. We have clinical trials happening in multiple drugs, and maybe soon we'll see some or one or two reaching the final line. And I think that we'll have to start thinking that the standard of care is perhaps about to start changing. So the designs that we are talking, and this goes to the placebo, may become a little more difficult. And I really enjoy the view of the gentleman over there, I'm sorry, I forgot your name already, that we do have a placebo arm, or maybe in an event from other trials, we have placebo arms. So not only on top of the clinical trials, we do have within trials placebo arms. Why do we need more? And I do appreciate all the consideration about the effect size and the endpoints, and I'm the least expert to talk about them, and methodologists are more clever than me on that. But I think we need, for rare diseases, to start thinking about using the data that they are generated for the benefit of patients. And I do like that, the idea of using already existing placebo arms. And the final point I want to make is, as I said, the effect, yes, different drugs will have a different effect. But for me, the other thing that is important, and it was captured as well by patient, is the duration of the trials. So if we see a miraculously turning of a milestone at patients not sitting or not walking and suddenly walking, that is very interesting, or the halting of the disease and the slowing down of the disease. But for me as a regulator, isn't it an important question for how long that will be? So these are children that we treated, let's say, from birth very, very early. When is the benefit risk going to change? Is it going to be like this? So for me, the duration maybe hasn't been discussed so much already, but it is an important factor if you take the safety profile in this. And of course, that will not be as part of a marketing authorization application. We're going to have a clinical trial running forever. But I think we need to reflect very, very careful and see, compared to the natural history, what is the benefit in terms of time, not just effect. Thank you.
I have my name is Francesco Montoni. I have a particular, not answer, but a consideration based on your aspect related to the duration of the effect. Clearly, I do not know the answer to that question. At the same time, we know from both the spinal muscular atrophy and the Duchenne field. From the spinal muscular atrophy, we know that the long-term effect is, or the long-term achievement of the long-term survival is depending on the maximal achievement the patient achieves. So I think that the patient who have achieved walking, for example, at any time, they will have a totally different trajectory from the patient who, that is irrespective of whether they lose the ability to walk or not. The patient who are able to sit unsupported, they have a completely different trajectory for the patient who never able to sit. So I think that there is at least from the natural history studies of this patient, some information that the maximum achievement determines the long-term trajectory. And clearly, in other conditions, such as in Duchenne, we do know that if you do move, for example, using corticosteroids, you do move the rate of deterioration or individual time point that has a very long-term effect in the respiratory failure at a later stage and death at a much longer, after a much longer point. So on one hand, I completely agree that it will be very nice to know how long this effect will be achieved. At the same time, the patient themselves are telling us the long-term outcome is really depending on what has been the maximum achievement, especially in spinal muscular atrophy. So from that perspective, it may be a very interesting long-term question, but in the short term, it may not be so crucial.
Hello, my name is Paul Moore. I wanted to just give a viewpoint as we weigh in and thank the regulators, is the viewpoint of the families. When Mencia and Elliot spoke about getting up 8 times at night, or when I think of pulling over on the motorway because you have to use a suction machine or something, the timing, this is a devastating disease, especially for SMA type 1. So I think our appeal is we want to use a scientific approach. We have fought against the miracle approaches. We've been watching wiggly mice and pigs. I have been for 13 years. We've come a long way. So we don't want to ruin anything. And we know one mistake could compromise what is coming down the road in the pipeline. It's very difficult to recruit families for the trials. So industry is risking funds, money. The families are risking the time and the capability of their child to be involved in another trial. So please help us streamline and not duplicate studies and things, because we are a family, according to the data this morning, 25,000 just in Europe. Here I see an international group. There is a level of cooperation unseen in this rare disease. So please keep in mind, when we're looking at the scientific data, I'm not a biological scientist, a computer scientist, which has nothing to do, but we all want a proper methodology. It's too late for me but there is so much hope. There's so much, the expectations are very high. So please help us find a point to filter through and focus. What can we do with the family associations, with an ethical approach, to bring these to the endpoint as soon as possible? Because for many families, there is no time. Every day, every single day, a SMA1 child passes away. So when we look at times of 1 year, 2 years, it just seems unbelievable. Going back to the discussions of the everyday life. So it's not just the patient, it's the entire community, the entire family. Sorry, I'm a little bit emotional because it's a very important day. I thank all of you, but please let's remain the focus on how we can streamline the whole process.
Thank you very much. It's time just to wrap up this session, this magnificent session. But before, Pavel have something from the public. As you know, we are in streaming and we have an opinion and just a comment from the streaming people that is up there.
Yes, thank you. So this is probably to provide also food for thought for the next session. We have a comment from a pediatric neurologist in Bristol, Dr. Majumdar, who says that probably it's time for the SMA field to consider alternative approaches. For example, re-enrolling all comers into a registry and then using the whole registry by putting everyone on different investigational drugs and investigating them using, let's say, time to event, and see, in that sense, actually using the registry or one pool of data to see which drug performs how, and also having a comparative data. This is just something that comes to us as a comment, but as I said, it may be considered, especially when we discussed in the next session, the later onset patients.